Hello and welcome back to my channel. So we were discussing the second chapter of our first unit, that is diversity in the living world, and the chapter we are discussing is biological classification, right? And we have done with its introductory part, the five kingdom classification, and also we have started our first kingdom, that is Monera. Okay, and in that Monera is divided into two main divisions, that are Archaebacteria and Eubacteria. Okay, I guess that much is clear, and we have already drawn its broad prospect or broad classification actually. That is, Monera is divided into to Archaebacteria and Eubacteria, and for the Eubacteria is divided into bacteria and cyanobacteria. Okay, so we have already discussed Archaebacteria. For that you need to watch the previous videos to get in link with this topic and with this chapter as well. So today we are going to discuss about Eubacteria. Okay, so let's get started. U means what? U is true and bacteria is the organism. Okay, so Eubacteria are the true bacterias. Clear? And they lack a well organized nucleus. Okay, as well as they don't have well organized membrane bound organelles like we have. Okay, so they lack membrane bound organelles. Clear? But they have a cell wall, and cell wall is a feature of plants. Okay, cell wall is a feature of plants, but the bacteria do have a cell wall. Okay, and they have for these, as I have already mentioned, that Eubacteria is divided into two, that is bacteria and cyanobacteria. Okay, we are going to discuss these two in detail, a little bit in detail. Okay, we are going to discuss with this structure of bacteria, as well as its respiration, mode of respiration, nutrition, reproduction, economic importance. We are going to take up all. Okay, so be with me and do like, subscribe and share my channel. And okay, I'm not going to repeat this now. Let's erase this first. Okay, now starting with the first, that is bacteria. Okay, so bacteria, let's take up the normal features of bacteria that we are going to discuss first. The bacteria are unicellular. Okay, these are unicellular, prokaryotic. Okay, they have cell wall. They have cell wall like the plants have. They are smallest free living organisms. Okay, and the smallest one known, the smallest bacteria known is named Dalster pneumocentis. Dalster pneumocentis, and Its size is 0.15 μm. Okay, clear? And about, now we are going to discuss about a little bit in detail now. So let's take types of bacteria. Its types of bacteria. So what are the types of bacteria means what are the shapes in which the bacteria are being found. Okay, so let me take it right. Okay, so number one are coccus. Coccus are oval shaped. Okay, like these are what? Coccus. Okay, they can be diplococcus, streptococcus, staphylococcus, according to the arrangement of these uh, oval shaped structures. Okay, these oval shaped structures are coccus. Clear? Second one is bacillus. Bacillus are something rod shaped. They are like these, like this. Okay, they are something like this. You can find them, the colony can be found like this. Okay, these are what? Bacillus. Number third is spirillum. Spirillum itself says it's a spiral shaped bacteria. Okay, and these are having flagella on its both the end. Okay, flagella which help them to move. Okay, now last but not the least, it's vibrio. Vibrio are of comma shaped. They are like comma, like this. Okay, they are like comma shaped. So these types of bacteria can be found. Okay, these are the shapes in which the bacteria can be found. Okay, now we are going to take up the generalized structure of bacteria. Okay, so let's take the structure. Let me enlarge this for you a little bit less. Wait, yeah, now this is okay. Okay, so let's get some bright color. Yeah, so let's start with flagellae. Okay, let's start with flagellae. Okay, flagella in short. If you are talking about singular one, that is flagella. If you are talking in plural terms, it's flagellae. Okay, so it's a long filamentous appendage. Why we are calling it as an appendage? Because it is being used in movement of bacteria. Okay, it helps in movement of bacteria. Okay, and this is divided into three parts, that is filament, hook and vessel. Okay, its body part is divided into filament. Okay, this one is the filament. Okay, then hook and the basal part. Filament, hook and the basal part. Clear? Next, it's a Actually, it provides a rotatory function and contains a protein named as flagellin protein. Okay, clear? Flagella are present on bacteria. Clear? Next, they are long, filamentous, and they are appendages which helps in movement of body of the organism. Clear? And this flagella is divided into three parts, that is filament, hook, and basal part, and this contains a protein called flagellin protein. Okay, clear? I hope this much is clear to you. Let me arrange this so we can, we'll get the other things clearly. Okay, next move to another small structures present on the surface of bacteria. These are pilus, or called as fimbriae. Okay, or can be called as fimbriae. Okay, these are short filamentous structure like flagella, but they does not actually help in movement, but they rather function as a you can say that they can function as an attachment of bacteria to surfaces. Okay, these help in attachment. Okay, they also contain protein called pilin, pilin, pilin. Okay, this is the protein contained by pilus. Clear? Also known as fimbriae, has a short filamentous structure, helps in the attachment of bacteria to surface, and contains a protein called pilin. Clear? Next comes the outer layer, the outermost layer, and the outermost layer is the capsule. Okay, the capsule is the outermost layer, and it is made up of gelatinous polysaccharide and polypeptide, composed of gelatinous polysaccharide and polypeptide. Okay, and it protects the bacteria from pathogens and desiccation. Okay, and also helps in the adherence to any surface. Clear? First thing, outermost layer is capsule. Capsule is made up of gelatinous polysaccharide and polypeptide. Clear? And what is its function? Its function is to protect the bacteria from pathogens and desiccation. Desiccation means from, get ruptured, getting ruptured. Okay, and it also helps in adherence to the surfaces. Yeah. Now move to the next layer, that is the cell wall. Okay, let me change the color, otherwise you're going to get confused what I'm writing in what respect. Okay, bad choice. Okay, let me take some other, this one. Okay, clear? So cell wall, the cell wall is the second layer of the bacteria, and we can also call it as a peptidoglycan cell wall. Okay, and it is actually made up of N-acetyl glucosamine, N-acetyl glucosamine, that is also known as NAG. Okay, and formed of two complexes. Okay, one is N-acetyl glucosamine NAG and another one is N-acetyl muramic acid, that is NAM. Okay, N-acetyl muramic acid, also known as NAM. Okay, this wall is made up of two, that is N-acetyl glucosamine NAG and N-acetyl muramic acid, that is NAM. Clear? So cell wall also contains magnesium ions which binds to teichoic acid in it to provide the stiffness. Okay, magnesium ions are also there which attaches to teichoic acid to provide stiffness. Okay, this binding protects the bacteria from thermal injuries. This binding protects the bacteria from thermal injuries. Provides stiffness, so it protects the bacteria from thermal injuries. Okay, hot environment, hot habitat in which they live, that can be protected from this combinations, from this from this binding. Okay, I hope this one is clear. Cell wall is made up of N-acetyl glucosamine and N-acetyl muramic acid. There are also magnesium ions which are being binded or which bind with teichoic acid, and these bindings helps the bacteria to these bindings protect the bacteria from thermal injuries. Okay, this much is clear. Flagella, fimbriae or pilus, or and the next outermost layer is capsule, that is gelatinous polysaccharide and polypeptide, made up of gelatinous polysaccharide and polypeptide. Then the next one comes the cell wall that we just have discussed, formed of NAG and NAM. Okay, now moving towards the next, that is the plasma membrane. Okay, plasma membrane is made up of phospholipids and proteins, that we all know. Okay, and it is a site of some respiratory enzymes as well. Site of some respiratory enzymes. Site of respiratory enzymes. Clear? Form of phospholipids and proteins. Plasma membrane is formed of phospholipids and proteins. Clear? So moving towards the next, that is the cytoplasm. Okay, cytoplasm contains 80% of water. Cytoplasm contains 80% of water, and then proteins, carbohydrates, lipids, organic ions are also present in cytoplasm of bacteria. Clear? Then understand, let's move towards the next one, that is the another point is the plasmid. Okay, this part, this structure which does not have actual shape, okay, which can variate in every bacteria. So these are the plasmids. Plasmids are somewhat small, they are somewhat circular, not clearly but somewhat circular, and they are self-replicating. Okay, they replicate by themselves only, they don't need any help of DNA and all. Okay, and also by which they'll replicate, they does not need nucleoid DNA, they need only their extra chromosomal DNA. Okay, so they are small, circular, they have self-replicating extra chromosomal DNA. Extra chromosomal DNA. Clear? These are the points of plasmids. And these DNA have genes. Okay, having few genes. Few. Clear? So plasmids are what? They are you can say that they are a extra chromosomal DNA which is being present at some which is present in cytoplasm, and they are self-replicating, and they are small, circular, and these DNA has few genes as well. Okay, so moving towards the next, and the next is the ribosomes. Okay, let me change the color to make it more interesting a little bit. So chromosomes are what? Chromosomes are small round like structures present in the cytoplasm of bacteria. Okay, they help in replication. Okay, they actually translate the RNAs, okay, to provide some products that are needed by the cell, protein in the form of protein. Okay, so ribosomes, these have 70s types of ribosomes. Consists of rRNA, ribosomal RNA and proteins. Clear? Next, moving towards the nucleoid. Let me clear this first. Otherwise, we are going to make a hodgepodge condition, which is not good for us, because this will lead to confusions what I am writing, whether I am going to clear the all colors or not. Though it's look amazing with different colors, but still, to make you clear everything, I need to rub this thing. Yeah. Now let's get started with the DNA, nucleoid. Okay, so nucleoid is the nuclear area. Okay, this is what the nuclear area, and it is amorphous lobular mass of fibrillar chromatin type material, or say genopore. Okay, which actually occupies 10 to 20% of cell. It's nothing but a mass of chromatin which is present in the cytoplasm of bacteria, which covers up around 10 to 20% of the cell area. Clear? And apart from that, they have some inclusion bodies, like here if I draw it, some inclusions bodies, and these inclusions, let me write it down to make it clear. Inclusion bodies. What these inclusion bodies do, they actually reserve food for the cell in the form of glycogen, starch, lipid and sulfur granules. Okay, and apart from them, there is a small fold-like structure. Let me change the color for that. Small fold structure that is known as mesosomes. What mesosomes do? It's just a folding. okay, which serves as a respiratory organ, serves as a respiratory organ, respiratory organ for the bacteria, okay, or we can say that it's the center of respiration. Mesosome, it's just a folding on the cell, it's folding on the bacteria. Clear? I hope this much is clear to you. In the next video we are going to take up the respiration in bacteria, because if I discuss this right now, the video will be much longer. Okay, so we'll discuss that in the next video about the respiration, nutrition mode and rest. Okay, thank you, and do watch the remaining earlier videos as well. Thank you very much.